Hello everyone, good morning. My name is Sanjay Suresh and I am a student at University of New England taking Biochemistry, Biomedical Science. Today I'll be discussing two important cases that I've encountered. The first case is I'd like to discuss about a newborn who appeared healthy at birth, and then within 24 hours it developed lethargy, hypothermia, apnea, and then when we did laboratory tests, it revealed that there was severe hyperammonia with the ammonia levels increasing from 314 to 600. So basically, with elevated orotic acid, urine orotic acid, and normal plasma citrulline. So I like to diagnose this as an ornithine transcarbamylase deficiency, or an OTC. Which is strongly associated with the urea cycle. So before I go into the other details, I'd like to talk about the normal mechanism of ammonia transfer. So what is ammonia? Ammonia is basically produced from amino acid breakdown, and it is highly toxic, particularly to the brain. So most of the amino acids are first transferred to alpha ketoglutarase through a process called transamination, forming glutamate. And this reaction is catalyzed by aminotransferases, which requires vitamin B6. And glutamate then plays a central role in nitrogen metabolism. And in the liver, glutamate dehydrogenase removes an amino group from glutamate and releases free amino acids that enter the urea cycle for detoxification. So in the extrahepatic tissue, excess ammonium is converted into glutamate by glutamine by glutamine synthase. And glutamine safely transports ammonium through the bloodstream to the kidney and the liver. And in the liver, glutaminase is converted back to glutamine, and then back to glutamate, and then ammonium. And then this ammonia enters the TCA cycle. So now I'd like to discuss about the nitrogen and how it's connected to gluconeogenesis and fasting. So in the muscle, the protein, basically they're broken down into amino acids, and most of the amino acids are transferred to pyruvate to form alanine, which travels to the liver and then through the bloodstream. And in the liver, alanine is converted back to pyruvate, which is used for gluconeogenesis to produce glucose. So basically, the amino group is transferred to glutamate, and then it is released. It releases ammonium into the urea cycle, and this is called glucose-alanine cycle. So, coming to the laboratory findings, marked elevated ammonium and high orotic acid levels and low citrulline is diagnosed as ornithine transcarbamylase deficiency. So normally this ornithine transcarbamylase, what it does is it converts ornithine and carbamyl phosphate and citrulline. And when this is deficient, carbamyl phosphate accumulates in the mitochondria, and it leaks into the cytoplasm. And this enters the pyrimidine pathway. So producing excess orotic acid helps to explain why the orotic acid levels are high. And another enzyme which is likely to be deficient is arginine succinate lyase, because this disorder typically requires high citrulline levels, and unlike normal and low citrulline in this patient. So for OTC deficiency, arginine becomes a conditional amino acid because of the blocked urea cycle cannot produce enough arginine. So therefore, dietary arginine supplementation is often given for the treatment, and this helps to support growth and provide residual urea cycle function and promote nitrogen excretion. So on the whole, this is diagnosed as ornithine transcarbamylase deficiency. So the next case that I took up is case 3, and this one is a case about bortezomib and the ubiquinone proteasome pathway. So here in this case, we have a 45-year-old woman with multiple myeloma who has been treated with bortezomib. And during this treatment, she develops increased protein accumulation and impaired cell signaling, and it leads to side effects. So what happens here? To understand bortezomib and how it works, it requires an understanding of protein degradation. So I'm going to talk about that here in just a moment. So our cells, they continue to break down old and damaged unnecessary protein through two major pathways. One, we have the ATP-dependent ubiquinone proteasome system, and then we have the ATP-independent lysosome pathway. So what is this ubiquinone proteasome system? So here, proteins that need to be degraded are first tagged by a small protein called ubiquinin. And this process requires ATP, and it involves three enzymes. That's E1, E2, and E3. And once this protein has been tagged with a chain of ubiquinin molecules, it is recognized by a protein called 26S proteasome, which is a large protein that unfolds, which is a large protein complex which unfolds proteins, and it breaks it down into small peptides. And this pathway is responsible for degrading short-lived regulatory proteins, damaged proteins, misfolded proteins, and it makes them essential for normal cell growth and cell cycle regulation and apoptosis. So now coming to the second pathway, which is the lysosomal degradation pathway. This is ATP dependent. So lysosomes contain digestive enzymes that break down extracellular, long-lived proteins and damaged organelles and cell surface receptors through processes called endocytosis and autophagy. So endocytosis means taking in. And together, these two pathways, they maintain the protein control and cellular homeostasis. So what does bortezomib do? It mainly targets this 26S by inhibiting beta 5 catalytic subunit, and then these proteins are tagged with the ubiquinone, which rapidly degrades the proteasome. And how proteasome blocks this activity is these proteins can no longer be broken down, and then they accumulate inside the cell. And this accumulation occurs in the endoplasmic reticulum, and this leads to stress and buildup of unfolded proteins. So if this stress becomes severe enough, these cells undergo apoptosis, which is also called programmed cell death. So coming to the case of multiple myeloma. So multiple myeloma is basically a malignant plasma cell disorder where we have large amounts of antibodies, and as a result, this heavily depends on the proteasome to eliminate these excessive misfold proteins. So by blocking this pathway, bortezomib overwhelms the cancerous cells with protein buildup, and it ultimately leads to their death, and it leads to elimination of the disease. So when we talk about the ubiquinone proteasome pathway, this also plays an important role in the immune system. So this proteasome mainly generates peptide fragments which present to the major histocompatibility complex class 1, and this allows cytotoxic T cells to recognize abnormal cells. So in addition, the proteasome regulates a molecule called NF-κB, which is a transcription factor. This is involved mainly in inflammation, immune response, and cell survival. So proteasome inhibition, this mainly prevents the degradation of IKB, and then this inhibits NF-κB molecule, and therefore this reduces the inflammatory signals, and this contributes to the drug's anti-cancerous effects. So basically, this is effective, and some side effects we can see is peripheral neuropathy, which results in protein accumulation, bone marrow suppression, which leads to thrombocytopenia and neutropenia. This patients can also have fatigue, nausea, vomiting, and constipation, and we can also have reactivation of herpes zoster. So in summary, the bortezomib, it works by inhibiting the 26S by preventing the degradation of ubiquinone target proteins, and this leads to protein accumulation in the endoplasmic reticulum and stress, and leads to apoptosis. So because this ubiquinone pathway is mainly essential for normal protein turnover and immune functions, inhibiting this pathway can also produce a lot of side effects. So by understanding this mechanism, it can help us, it can explain both the therapeutic effects and toxicity of this bortezomib therapy. So thank you. I hope you liked my video.